Our final presentation for session 1 comes from Dr. Hao Zhu. He's the director of the Division of Pharmacometrics within the Office of Clinical Pharmacology at the FDA, and he's presenting the FDA landscape analysis. Thank you, Dr. Zhu.
Well, thanks for a nice introduction. I would like to thank the organizers for providing me the opportunity. I'll be happy to share some of our thoughts and experiences on the use of artificial intelligence or machine learning in drug development and improve precision medicine. Before going into the details, I have a disclaimer. What I'm presenting here today are my personal views and should not be considered as the official position of the US FDA. My presentation includes the following sections. I'm going to start with an introduction on general concepts, the relationship between model-informed drug development, or MIDD, and machine learning, and the review function of AI and machine learning models and the Office of Clinical Pharmacology and FDA. Next, I'm going to share our landscape analysis results for AI and machine learning related submissions to the FDA in the past few years. Then I'm going to focus on some of the case examples we have seen in recent submissions and one emergency authorization review for Anakinra. After that, I will share some of the ongoing research activities and the AI and machine learning team. Last but not least, I'm going to conclude with some take-home messages. Well, let's begin by looking at what is artificial intelligence and machine learning approach and how it is different from traditional pharmacometrics models, which we used to describe, for example, exposure-response relationships. We know that there is a slight difference between the concept of artificial intelligence and machine learning models. For the sake of simplicity, we're going to use the two terms interchangeably in our presentation today. Machine learning is a field of study that gives computer the capability to learn without being explicitly programmed. During the machine learning process, a computer program is learning from experience with respect to a task. If its performance and the task improves with more experience. If we think about experience as data, this definition implies that the performance of the algorithm improves when it is presented with more data. Then I'm going to compare the general difference between the statistical model or pharmacometrics model with the machine learning model. In general, the traditional statistical or pharmacometrics modeling uses explicit model structure defined with domain knowledge and assumptions, while the machine learning model is very flexible and uses a very general model structure with less reliance on domain knowledge and assumptions. Second, the traditional statistical or pharmacometrics modeling tends to excel in handling data with a small or moderate number of independent variables, while machine learning approach are good at handling data with a large number of independent variables. It can also handle new types of data such as medical imaging. Third, the traditional statistical or pharmacometrics modeling is easier to interpret or explain or draw statistical inference, while the machine learning approach, such as artificial neural network, are harder to interpret or draw statistical inference. Instead, machine learning is more focused on prediction performance. Next, I'm going to talk about the relationship between model-informed drug development, or MIDD, and AI machine learning models. MIDD refers to the concept of development and application of exposure-based biological and statistical models derived from preclinical and clinical data to address drug development or regulatory issues. As you may see in the figure, the MIDD toolbox includes various modeling approaches. For instance, pharmacokinetic models are broadly used to support clinical pharmacology aspect in new drug development. Commonly used models include population PK models and physiologically based PK models. As an extension, exposure-response models are used to link exposure changes and changes in biomarkers and clinical outcomes. In the past few years, disease progression models are developed to describe the natural disease progression and various patient characteristics that may attribute to the disease progression. Clinical trials may be simulated through in-silicon trial simulation in order to optimize some of the design components. In recent years, QSP models are used to provide the current mechanistic understanding of the underlying disease and pharmacology by linking various biomarker changes and potential clinical outcome changes in the same platform. AI and machine learning models are the newest addition to the toolbox of MIDD. It has been shown to be a powerful tool to understand patient heterogeneity in disease progression and drug response, and therefore provide a unique angle to improve precision medicine. Potentially, AI and machine learning models can be applied to inform enrichment design, improve patient selection, manage safety risks of patients enrolled in a trial, optimize dosing for patients and different patient subgroups, and identify appropriate treatment for different patient subgroups. And we believe that AI and machine learning as a new handy tool for MIDD will be able to bring drug development and precision medicine into a new level. We're very excited to share the great news that the Office of Clinical Pharmacology has built up the AI and machine learning review function. Our primary interest is to apply AI and machine learning tools to improve our understanding of heterogeneous patient population in terms of disease progression and responses to our treatment. We believe that AI and machine learning tools can provide us more opportunity to enhance precision medicine. This slide shows you the main team members. I'm going to share with you some of the review and research projects conducted by this team later on. As a team, we would like to work together with the community to fully harness the power of AI and machine learning models to improve precision medicine for the benefit of patients. Next, I'm going to provide a high-level summary for AI and machine learning related submissions and the FDA. Actually, we conducted landscape analysis for the application of AI and machine learning in regulatory submissions back in 2021. The findings are published in Clinical Pharmacology and Therapeutics in June 2022. You may see the general information on submissions and some specific examples in this manuscript. This presentation will provide you an update from the existing publication by including the applications submitted up to December 2022. This figure summarizes yearly AI and machine learning related submissions to the FDA from 2016 to 2022. We can see a drastic increase in the relevant submissions in the past 2 years. There were only 1 or 2 submissions per year before 2019. Around 2019 to 2020, there were approximately 10 submissions per year. In the year of 2021, the number of submissions was increased to more than 130. Over the year of 2022, the number of submissions was way above 150. From the year 2016 to the end of year 2022, there are more than 300 submissions containing AI and machine learning components. This figure provides a summary on the distribution of the focused development stages for all the submissions. We notice that using AI and machine learning for drug discovery is extremely active. However, the low level of submissions in drug discovery is not surprising because FDA is not actively involved in the drug discovery stage. Instead, AI and machine learning approaches have been used in some of the submissions on repurposing of existing compounds for alternative indications. There are reported usage of AI and machine learning approaches to understand potential toxicity of a new compound under development and preclinical stage. However, vast majority of the submissions are at clinical stage, with a few submissions centered around unresolved issues after the compound is approved. Among all the AI and machine learning related submissions, about a quarter of them are in oncology related fields. Other common therapeutic areas include psychiatry, neurology, gastroenterology, hepatology, and medical imaging. In the year of 2022, we started to see the use of AI and machine learning for new drug development in rare diseases and hematology. AI and machine learning approaches are applied to predict outcomes, handle confounding effect, and select covariates. Some machine learning components are included in the typical pharmacometrics modeling. In addition, AI and machine learning models are used to detect anomaly, handle imaging, video, or audio data collected in the trial, or phenotype patients with real-world data. In addition to use in drug discovery and preclinical stage, as we mentioned earlier, most AI and machine learning models are applied to support clinical drug development. For example, it is used to enrich patients, stratify patients with different risk factors, select or optimize dosing, or ensuring adherence to the assigned dosing regimens. Furthermore, there are attempts to use AI and machine learning models to select endpoints, identify biomarkers, or synthesize control groups. Now I'm going to move on with some case examples we have seen in the submission. The first example is for biomarker and endpoint assessment. In this case, an AI algorithm is used to evaluate medical imaging-based biomarker and seek the application of this biomarker as a potential endpoint for future clinical trials. The second example is for patient selection. In one submission, an AI-based diagnostic biomarker is used in conjunction with clinical assessment to identify patients and a defined disease stage. This approach is considered as a part of the inclusion criteria. In another submission, a companion diagnostics is developed by using an AI algorithm linking EEG, digital biomarkers detected through wearables, and other clinical symptoms. And this approach is used to select appropriate target patients. Next, I'm going to focus on the recent review case where we applied a machine learning algorithm based on patient baseline characteristics commonly collected in clinical practice to select the patients. The drug is called Anakinra. It is interleukin-1 receptor antagonist indicated for treating patients with rheumatoid arthritis, cryopurine associated with periodical syndromes, and deficiency of interleukin-1 receptor agonist. The sponsor is seeking emergency use authorization of Anakinra for certain hospitalized COVID-19 patients. The main basis for this application is a clinical trial called SAVE-MORE study. This trial was a randomized, double-blind, placebo-controlled study to evaluate the safety and efficacy of Anakinra in patients with COVID-19 pneumonia who are at the risk of developing severe respiratory failure. The COVID-19 pneumonia should be radiologically confirmed by chest X-ray or CT. More importantly, all enrolled patients in the study are required to have a plasma-soluble urine kinase plasminogen activator receptor, or known as SUPAR, level greater than 6 nanogram per ml. The results suggest Anakinra is efficacious. However, SUPAR assay is not commercially available in the United States. This formulates our review question. That is to search for alternative score based on commonly measured patient characteristics to identify patients with SUPAR greater than 6 nanogram per ml. The sponsor provided their initial proposal. For our team, we assessed them and used a machine learning classification model with elastic net regularization to identify additional contributing features. In addition, a new network-based model was used to independently assess the importance of the additional feature and its cutoff value. Both models suggest that additional criteria of neutrophil to lymphocyte ratio with a cutoff value greater than 7 should be included. In addition, we identified the relevant evaluation metrics to assess the performance of the selected scoring rules. To us, the most critical metrics should be positive predictive value, or PPV, which indicates the likelihood of patients being super high once they are selected by the identified scoring rule. Specificity is also important as it shows us the chance that patients being rejected by the scoring rule indeed are super low patients. Another relevant factor is sensitivity, which shows the likelihood of super high patients being selected by the identified scoring rule. We also considered the mechanistic plausibility for the identified features. The model performance using the finally selected scoring rule is shown in the two tables below. The table to the left is the results from the training dataset. The table to the right shows the results from a test dataset or an external validation dataset. The information in the test dataset is obtained from SAFE trial, which is totally independent from the trial used for model building. The PPV value from the training dataset, but more importantly from the test dataset, is very high, which is about 94%. This ensures that once patients being selected by the scoring rule is likely to be super high patients. High specificity, reasonable sensitivity, and biological plausibility ensure that the selected scoring rule can be reasonably applied. Based on the final selection, the current fact sheet contains the following language. Anakinra is authorized for emergency use for the treatment of COVID-19 in hospitalized adults with positive results of direct SARS-CoV-2 viral testing with pneumonia requiring supplemental oxygen who are at risk of progressing to severe respiratory failure and likely to have an elevated plasma suPAR. The fact sheet also explains that suPAR assay is not commercially available in the US. Section 1.1 of the fact sheet includes the information on the identified scoring rule. A total of 8 items should be considered. Patients meeting at least 3 out of the 8 criteria are likely to have a suPAR greater than 6 nanogram per ml, and this helps to ensure that appropriate patients will be selected with good alignment with patients being tested in the clinical trial. With that, I'm going to switch gear to talk about the research projects. We have several ongoing projects and are looking for fellows. If any of the audience is interested in, please apply for the position through the provided link. In OCP, our research projects target different directions. We conduct a landscape analysis to fully understand the current status on AI and machine learning related submissions at the FDA. We also explore novel methodologies that can be applied to improve our understanding of heterogeneity of patients and enhance precision medicine, or to improve new drug development. In addition, we try to apply machine learning models into therapeutic optimization and individualization. Several projects are undertaken in the 3 directions, and some of the project results are published while some others are still ongoing. We'll list our recent publications in case you are interested in. In the following few slides, I'm going to provide a high-level summary of 2 projects as examples. The first project is designed to improve clinical trial design. As we know that some of the clinical trials face the challenges of placebo responders, which may attenuate the detectable efficacy signal and therefore decrease the chance for trial success. This project is intended to identify patient baseline characteristics that can be utilized to predict placebo responders by using AI and machine learning models. Then we attempted to apply the selected patient characteristics as additional exclusion criteria, followed by evaluating the treatment effect after additional patients being removed. The preliminary results suggest that the treatment effect can be numerically increased after patients with baseline characteristics associated with a large placebo effect being removed. And this finding may provide additional insights for future clinical trial design. The second project is to improve precision medicine. We understand that certain treatment may lead to rare but severe adverse events. We attempted to use AI and machine learning tools to identify patient characteristics that can be linked to the incidence of the rare adverse events. In order to validate the findings, we try to compare the results through clinical trial data and real-world data. We're in the hope that identified characteristics for high-risk patients may inform appropriate selection of therapy and risk mitigation plan. With that, I'm going to conclude my presentation with some take-home messages. AI and machine learning as a new addition to MIDD is a powerful tool to improve drug development and patient care. The application of AI and machine learning to support drug development is expanding rapidly. With the capability to link various patient characteristics, for example, demographic information, vital signs, and lab measurements, AI and machine learning tools may provide additional insights on precision medicine. I would like to acknowledge a long list of individuals who have helped me to prepare the presentation. In addition, I would like to thank the contributions from the review team, the research project team, division members, office members, and our fellows. Last but not least, I would like to thank you, our audience, for your attention.